it is so nice to see so many people here. We are, on behalf of the HPA team, we are so excited to launch this version 24. It's a very major update. And what we will do is that we will show a lot of data to give you a glimpse, but obviously the most important is that you go into the protein atlas and do your own things afterwards. The version 24 was released just a few minutes ago, so it's online, but you're not allowed to watch it until you have finished this lecture. So I will say a little bit about the first 20 years, but then I will also go through all the new features in the version 24. Then Cecilia, that some of you might have heard yesterday, will talk about the tissue resource, and Emma, that had her brilliant talk this morning, will also give you a glimpse of the subcellular resource. And then Maria Bueno Alves will talk about the new disease blood atlas, and then I will finish up and tell you a little bit about the new knowledge summaries that we are introducing today. Because the first version of the protein atlas was actually done at the UPO meeting in Munich, organized by Matthias Mann, who is in the audience. So we have now had funding from the Wallenberg Foundation, which is a non-profit organization in Stockholm, for 20 years. And that has allowed us then to build this database in the way that you know. We want to ask very central questions of human biology and diseases. How many different proteins are expressed in the cell, and what are the specific proteins and so on. But also, what are the proteins which will be the targets for future diagnostics and therapeutics. So this is, of course, very important for human biology, but also for medicine. We are very proud that we generated actually about 12 million images, which means that it is one of the largest efforts to generate tissue images in the world. Also recently, we have some key publications. I just listed some of them on this. The single cell in Nature last year, the colorectal paper that just came out a week ago in Nature, and the spatial transcriptomics, which is collaboration with people at BGI in China, that came in Cell also last year. We're kind of proud that we are sort of having 500,000 visits per month, and this is going up, as you can see, and this is of course fantastic to serve the community in this manner. This is an interesting map. It shows the distribution of visitors around the world, and you can clearly see that medical research is done in the United States, of course, in Europe, of course, but also in Southeast Asia. And then it is a little bit blank in other parts of the world, but we hope that they will also catch up. We have sort of divided this protein atlas journey into sort of 3 different parts. The first one was very much to make antibodies to all human proteins and analyze those in tissues and cells. The second decennium was very much about integration of omics, where we started to use these fantastic tools on transcriptomics, single cell RNA, the RNA-seq, and also lately spatial transcriptomics. But now we're moving into a more data crunching era, where we hope to then use data-driven life science to actually try to understand human biology even better. We're also very proud to be core research infrastructure, so we're very indebted to LXC that has helped us a lot. We are then selected as a European core research. We also recently was selected by the US-based Global Core Biodata Resource to be one of their core resources, and obviously this is very, very nice for us. So in summary, we have during the years generated and actually annotated 5 million web pages, which are annually updated, and we get a lot of updates today actually. We just a few minutes ago launched the version 24, and I thought I would just quickly go through this before I hand over to Cecilia Emma and Maria. So there are some major updates. One is that we are sort of trying to make it a little bit more easy to navigate. So we are going from 12 sections into 8 resources. We have a lot of new data, and I will say a little bit about that, especially on the single cell side. We have new knowledge summaries. I will also talk about that. We are very excited to launch for the first time a first part of a pan-disease blood atlas. This will be followed by several other stages later this spring, in the spring and maybe next year. But we're also trying to adapt to our AI type of work by having increased downloadable data availability. So first about the 8 resources then. So the tissue resource, and Cecilia will say a little bit more about that, but one of the new features that is that we have added now even more tissues to the multiplex tissue profiling. This gives you wonderful, wonderful images of the human cell and the tissues, as you can see here, but you will hear more about this very soon. The subcellular resource, I almost doesn't have to introduce to you, since Emma did an excellent job this morning, but you will also hear a little bit about her. We have the focus this year on primary cilia, the sperm structures, and also the metabolism variability. One of the big things this year is the blood resource. We are focusing now, and we are launching 59 major diseases done with Olink Explore, and also a lot of healthy, 6,000 patients, and we are releasing data, open access for 1,100 proteins. And you will hear more from Maria about this. Brain resource is another resource, and here we also have a new feature with the spatial transcriptomics. This is a collaboration with BDI in China. We had a paper in Cell in 2022, and the data from that in the human cortex is now released, and it's a lot of data, so I hope you can explore that. This is the sort of images that we can generate. This is actually from cerebellum. And you can actually see, this looks almost like an immunohistochemistry build, but this is actually RNA data. It's single cell resolution, and in every cell here we have a data for all the 20,000 genes. So it is quite amazing. It is a revolution for all of us working with transcriptomics, and you will of course hear more about this in the future. The cancer resource, we have added validation cohorts, but we also have imported data from CPTAC. Henry Rodriguez talked about this earlier, and we're very happy then that you can compare the mass spec data with the antibody-based data and the RNA data. The cell line resource, we also have imported MS data. Here we have now data for more than 1,000 human cell lines, and this is, I think, a very good resource to make for researchers that wants to use cell lines for their experiments. The resource that is changing more is the single cell resource. It's a lot of new data. And obviously part of that is that so much is being produced around the world, and we are importing a lot of data from others. So we are very indebted to that we for both the Topla Sapiens that has and the Cell Atlas that has made a lot of nice data that we imported, but also we have on the brain cell type, we have also single nuclear data that has been generated at Karolinska. So the Topla Sapiens and the BioHub from San Francisco, this is then integrated into our data, and you can compare then our own generated data with the data from Topla Sapiens. And then last but not least is the structure and interaction resource. So we have more than 80,000 structures being visualized. And it took us 3 months of CPU time in a supercomputer center to do that. So then you can compare the structure of the sort of consensus protein, the full length, with the structure of the splice variants. And then the interaction resource, we are very indebted to EBI and Intact and Henning and others, but we have also added data from BioGRID, OpenCell and Bioplex. So with that, I'm going to hand over to Cecilia, and she will quickly say a little bit about the tissue resource. So here you are.
Thank you very much. So the tissue resource builds upon this flagship paper that we published in 2015, where we looked at protein localization across all major tissues and organs, and then we mostly used immunohistochemistry and bulk RNA sequencing to classify the genes according to tissue specificity. But now we want to go one step further and go more into detail in different cell types and organs. So the way we do that is that we have built a high-throughput multiplex profiling workflow, and there we look at single-cell protein profiling using an iterative fluorescent-based, antibody-based imaging workflow. And we're also adding data from high-plex multiplex profiling, where we used more than 40 different markers that can be used as a benchmarking data set for image analysis. And this part you can find as part of the dictionary, where you can learn more about the different tissues and organs. And the way we have been doing that is that we used an iterative fluorescent-based workflow. So we used the fixed panel markers that were built upon single-cell RNA sequencing data, and then we replaced one marker in each run for the candidate protein that we want to investigate. And then we can look at overlap in expression with these fixed markers. And that then allows us to look at single cell types, but also subsets of cells and subcellular structures. So I'm going to introduce the different projects that we had using this workflow. And we used 7 different antibody panels for 8 different tissue types. So we have been studying in total more than 1,000 proteins using this type of workflow. The first one that we spent most time on is testes, because it's a very complicated organ with lots of cells that differentiate throughout spermatogenesis, and some of these cells are not possible to distinguish by the human eye. So here the antibody panels with multiplex really helped us to identify the protein expression in certain subsets of cells. So in total we looked at 13 different cell types and more than 600 proteins in testes. Here's just one example of Nanos3 that we, based on this methodology, could see that it's expressed in spermatogonia stage 2 and 3, which is a subset of the cells in the seminiferous ducts, the most basal ones. Then we also built a panel for ciliated cells, where we wanted to look more at the ciliated structure in motile cilia. So we developed a panel that outlines the different parts of cilia, so the nucleus, cytoplasm, and then also the transition zone, basal body, and axoneme of the actual cilia structure on motile cilia. And here we looked at 5 different tissues that harbor motile cilia in the human body, both female reproductive organs and also the airways. And there we can also study how these different ciliated proteins are maybe altered in terms of subcellular localization between different organs in the human body. And here's an example of a protein called CROC2, which we see that it's mainly localized to cilia transition zone and the axoneme that we see here in green. And then we also did multiplex profiling of kidney, and here we have built a panel that outlines the different tubules and compartments of glomeruli. So here we can, for example, see a protein which is mainly localized to distal tubules that you see here in green that overlaps very well with the red channel. And finally, we also built a panel for salivary gland that harbors different types of ducts, both large and small ducts, and different types of glands. So here's an example of an unknown protein that was not described before. So it's an ORF protein that we here could see that it's mainly localized to mucus glandular cells in salivary gland. And with a highly multiplex data set that we have been adding as part of the dictionary, we have used 47 different oligonucleotide barcoded antibodies. And that then allows us to toggle on and off all of these different channels. And you can also change the intensity of the signals. You can see where all these different specific cell type markers are expressed throughout all our normal and cancer tissues. So all the arrays that we have been using for immunohistochemistry, you can look at all these different markers, and this can then be used as benchmark data sets, especially for image analysis. And here's just a list of all the markers that are present in this panel. So I really hope you find this new data set available, and we're happy to take any feedback, and we will continue to add similar types of data for other tissues in the future. Thank you.
Hello, great to see so many of you here. I will try to keep this very brief because I think a lot of you heard me talk about it this morning. What I do want to say is that I shouldn't be getting the credit for this. It's really Ulrika. She's sitting over there. Ulrika, raise your hand. She's actually the hands and the brain and the heart behind the Subcellular Atlas. We show that 40% of all enzymes show single cell variability, and more than half of all enzymes localize to multiple subcellular compartments where they might perform different functions. So lots of biology to discover there. What is new is actually that we've assembled this data and made this landing page and wrote a paper so that it's very easy for you to access this data and start to explore it. The actual data has already been there for years, but that doesn't mean that people find it. So that's why we love these landing pages. The new structures, the cilia, this is something we worked very hard for. And as you heard earlier today, we've mapped proteins to the flagellum of sperm and also to the primary cilia of 3 different human cell lines. The sperm data is really beautiful, as you can see here. They look great on picture, and we can localize proteins to a lot of structures that other cell types don't have. So it's a really rich resource, although most proteins were known to be in sperm already. The cilia is much trickier. It's actually very hard to see on images. We've added arrows to help you find it because the images aren't always as beautiful, but the staining is super interesting still. So here we have lots of new cilia proteins and also spatial information about how they're distributed within the cilium. And again, the conclusions for the cilia is that individual cells can fine-tune their ciliary proteome, and this is substantially driven by protein trafficking rather than gene expression. And it seems like cell types have highly kind of adapted the proteins that they have on their cilia to suit their purpose. So altogether, we have the primary cilia of the sperm. Matthias showed this already, metabolism, and then also the multiplex image data set that Cecilia showed you that also are lab produced. So with that, I would like to hand over to Maria.
So a lot of the data that we are releasing today is plasma proteomics data, and it's mostly based on antibody profiling, but Matthias will show that there's also some mass spec. So the technology that we're using mostly is Olink, proximity extension assay, and this is because it's suitable for low abundant proteins, it has high precision, we can measure thousands of targets, and it also needs very low sample volume. So it's ideal for our application. We started with profiling healthy individuals in 2020. We have moved on to studying some diseases, but now in this era, as Matthias described, what we want to do is more integrative approaches, and we have already released a pan-cancer dataset back in 2022. And today we're here to release a bigger dataset, a larger amount of data comprising different disease types, from cardiovascular to cancer, infectious disease, and much more. So please browse through these disease types and find your favorite. And we are releasing over 1,000 proteins, and we have also performed different types of analysis, including differential expression and machine learning. And this is how the data looks like, the kind of plots that you can see at the Human Protein Atlas at the moment. And this is an example for IL-6, where you see very nice patterns in infectious disease, and it's 6,000 individual data points. So this is called the Disease Atlas Resource, and it's a lot of people behind this effort, of course. And the idea is to create an open access resource. We have started with proximity extension assay, also targeted proteomics, but we are expanding also in terms of technologies. There will be some more data released later next year. And we want to, of course, try to identify biomarkers that relate to each of these diseases. And we can not only look at specific biomarkers, but we can also explore which ones are shared across diseases. So that's the power of this data set. And we can also compare these diseases to healthy patients, but also to a very mixed background of diseases. This is a snapshot of our data. It's going to be released as a publication very soon, we hope. And you can see that the data shows how samples tend to cluster according to disease, and also diseases that are related tend to be grouping together as well. So another nice feature of our data set is that it spans many different age ranges, so we can look at the protein profiles across age. And this is just to encourage you to go to the disease atlas page, look at the disease summaries. We put a lot of work into making this very comprehensive. And we can also, of course, you can also look at these profiles across the diseases. So please go into it and enjoy. This is just a conclusion slide saying how we have profiled all the samples, we have run different types of analysis, and you can now go on and do some fun disease studies. Back to Matthias.
So thank you, Cecilia, Emma, and Maria. So I just want to say a little bit also about target proteomics, since this is a proteomics meeting. So we have decided to also to do target proteomics of all of these blood samples. And this is a technology that was developed and we published, Matthias Mann and myself, a few years ago together with Emma actually. Where we actually used standards, heavy label standards, to get absolute concentrations. I'm not going to go into the details here, but it is a fantastic way to standardize the samples and get very low CVs. I also, before I end, or we end, will say a little bit about the knowledge summary. So this is a new feature. We've had what we call landing pages before, but we are now sort of more trying to actually have pages where you can explore human biology and the human proteome. So this year we are releasing 16 of these knowledge summaries, spanning everything from the human secretome, the human membrane proteome, but also the beautiful results that you heard from Emma and Cecilia and Maria actually. We also have been focusing on trying to define what is tissue and cell specific of the proteins, and we have a knowledge summary page where you can really look at the overlap between what is specific in the tissues and what are specific in cell types and single cells, and this of course gives you added information. So to the left there, you can actually see an example of a protein where the tissue resource says that this is a retina protein, and then you go to the single cell resource and you see actually it's very specific for the road photoreceptor cells. So you get sort of increased resolution, obviously, by going to the cell type. This is another kind of typical. Here you say the tissue resource says that this is not that much of a specific protein. It is expressed in many, many different tissues, but when you go to the single cell resource, you see that it's very specific for endothelial cells. And obviously we have endothelial cells all over, so it's not so strange. So you get added information by combining these two. As Cecilia was saying also, we're quite proud to have a fantastic now tissue dictionary. It should be used by all of you that are teaching students. It has some stunning images and it's really quite fantastic for education. Also, one of the things that we're trying to improve on, I think we've been pretty good also before, but it is the data availability. So all the data is now in XML format, it is downloadable, it is open access with no restrictions, and also we have a collaboration with the EBI and the bioimage archive, so all the raw figures has actually transferred to EBI, so you can download them from there. It is 300 terabytes, so it's a lot of data. So in conclusion then, we're very excited to launch this new version. We have actually done quite a lot of major launches throughout the years, trying to add things all the time to the Human Protein Atlas. And what is spectacular from our side this year is the spatial transcriptomics, it is the pan disease, and we will add also new data to these two new resources. One of the things that we also think is quite nice is that you can do aggregated visualizations which are just for you. So you can do search results, get some genes, and then actually get bar charts and box plots and so on for those, your favorite proteins, and compare them with each other, which is quite nice. But also I think the new disease blood atlas, I think a lot of us are interested in moving into precision medicine and so on. So I hope that this will be contributing to these kind of efforts to try to actually save patients in the end. So what have we learned during these 20 years? Well, we have defined the housekeeping proteins. We're still doing that. We pretty much know what proteins and genes are specific, both in tissues and single cells. We have been working quite a lot on the secretome and defining that. It's also interesting to look at the drug targets which are out there and how they are expressed in the human body. As Emma was talking about, also we're very interested in metabolic enzymes, and also the specific proteins in different tissues. And we have this spectacular tissue which is testis, that has so many testis specific genes. I think the challenge now for the future is that we all should be, and I think one of the nice things about this kind of resource, which is open access and the data is downloadable, is that we can use it then to try to improve the knowledge about the human biology, and maybe even in the future do modeling of it using AI and other techniques. So before I end, I just wanted to say that I'm very grateful to the Wallenberg Foundation. This is a non-profit organization in Sweden that has funded us now for 20 years and is funding us for another 6 years. So this is of course very nice, but I also want to thank Elixir. It's also funding this effort. There's a lot of people that have been involved in this. This is just some of the senior people on this slide, but actually it's been more than 700 researchers involved in this. Most of these have not been paid by HPA. They have just participated by their own will, and we are of course very grateful for all of these people. So this is just a picture of the Human Protein Atlas. Before I end, I just wanted to I just want to talk a little bit about this guy, Shahab Adori. He received just a few weeks ago the Lennart Nilsson Award, which is a very prestigious award in Sweden. He's an HPA, or has been an HPA researcher, and he's been working with a fantastic technique called iDisco, where you can actually move around in the body with the use of antibodies and actually see the 3D parts of your inner cells. And so I want to show some images that he has generated, but if you're interested in the Protein Atlas, there is a tab called Educational Videos, and there's 18 videos there, and they are all stunning. So this is the movie. This is not an animation. So this is actually the real thing you're now seeing. So please enjoy this.
Deep down in us, we find what makes us function, what makes us human. Our proteins. And now, we see them like we've never seen them before. When we explore the world in new ways, we discover new things about us, about life itself. And now we see our proteins like we've never seen them before.
Thank you. So I hope you find these incredible journeys into the body fascinating. I do. And please go to the educational videos and look more at them. So with that, I'm thanking all of you to come to this event. We are very proud and been working very hard to make it as useful as possible for researchers around the world. And with that, then, let's end this. I think we will stay around so you can ask us questions, but of course we now soon has a banquet. So thank you very much. Thank you.